Good morning, everybody, and thank you so much for the introduction. I'm super excited to be speaking at this workshop and be on stage amongst many distinguished speakers that are heroes of mine from my PhD studies. So super grateful to be here. I'm thankful for the organizer and for putting up a great workshop that spans a spectrum of topics. Some are more on the biology side, some are more on the ML side, and this one for sure will be more on the ML side, so you've been warned. So to motivate this talk, so I'm going to talk about models for fitness prediction, and just to mention a few important applications of why we should care about fitness prediction models. The first one is human variant annotation, and the two prior talks from Dave and Megan already gave a great couple examples of why that is important in practice. Viral escape prediction is also another important application. If we want to anticipate the course of viral evolution during a pandemic, it's helpful to understand which mutations are going to preserve the fitness of viral proteins. And last one but not least, when we design novel proteins, we want those proteins to maintain function. And so it's very important to understand those fitness landscapes for those. As we develop models for fitness prediction, it was important to put together a benchmark to evaluate those various models that came up in the last 5 years. And so we put together this with many people, several of them are in the room, this benchmark of ProteinGym that builds on a curated collection of Differentiable scanning assays and clinical data sets. But someone was asking me yesterday during the workshop, what's the difference between ProteinGym and MaveDB? I think like the overlap is this data set component, but what makes ProteinGym is all of the other aspects. So like the fact that we've implemented different models and evaluated those on those different data sets, various performance metrics that evaluate those models depending on the context that you're interested in. If you're more interested in general fitness prediction, or if you're interested in protein design, you may want to look at different metrics. And also looking at like analyzing those models in different contexts, depending on the depth of the alignments, depending on the taxa, the mutational depth. Lots of great insights emerge from this analysis. Just to single out one of them is this curve that plots the size of a protein language model on the x-axis versus the average performance on ProteinGym. So one insight that emerged from this analysis is that there's a plateau in performance as you scale those protein language model, which was not obvious to begin with. I think there was like this hope in the field that as we were going to scale further those PLMs, we would continue to improve the performance, and we already reached a clear plateau, at least based on those Differentiable scanning efforts. Another important aspect is, and that's what I'm going to be focusing on primarily in the first half of the talk, is the importance of retrieval. And I think it's important to perhaps discriminate between two philosophies of building models. So there's the approach of training very big language models on larger databases like UNIREF 100. And when you do this, the models, they simultaneously do two things. They learn general patterns, biochemical features that are common across multiple protein families, but they also compress the information that is specific to each protein family. And I think that like the two philosophies are either you do this, and to be able to properly compress all of that information, you need a lot of parameters in your models to be able to remember and to remember all of that information. The advantage is that it's very convenient afterwards to use those models because you can forget about the underlying data. You can just plug a new sequence that you're interested in, and those models will give you a prediction. Versus the other approach is to use a rather lightweight model that is trained, that knows how to reason on retrieve sets of homologous sequences, and that's can broadly characterized as retrieval models. And I'm going to present a bit like introduce a bit like the current state and some recent developments that are making progress in that direction. So a first model, and that's this transception model that was being shown here on this slide. With a bit of retrieval, you go from a model that is sort of in the middle of the pack here in gray, and make it state of the art and like much better than models that are orders of magnitude bigger. And like you do this with a very simple approach, which is you train a protein language model in the standard way. It's an autoregressive language model in that case that is going to make prediction about the next amino acid in the sequence based on its prior context. So the first few tokens condition the probability of the next token in the sequence. And you complement this prediction by retrieving a set of homologous sequences to the wild-type sequence that you're interested in. And you look at the empirical distribution of amino acid at that position. And that gives you an empirical distribution that you can fuse with the autoregressive prediction. And by doing this, you get the nice boost in performance I was showing earlier. And so, like to step back and look at bucket different models in different groups in terms of the sensitivity to retrieval or how they deal with retrieval. So that the first generation don't use any retrieval at all. Second type of second category of models, they use MSAs or sets of homologous sequences somehow, but they are fixed set of MSAs. So you retrieve those MSAs with your favorite alignment-based approach, and then you train a model somehow on those MSAs, or you retrieve that, or you combine them at inference, as I was just mentioning in the prior slide. And the third paradigm that is emerging that I'm going to focus on in the next few slides is this notion of differentiable retrieval. And to motivate this, I wanted to talk about first the standard workflow to build alignment-based models. Step one, you have a retriever that can be, for instance, Jackhammer or MMSeq. So you plug a sequence of interest, and you're going to retrieve homologous sequences of that query sequence. And then once you've retrieved this set, you train a model on that set of sequences. And that has been leading to some great results in prior years. This is the backbone of models like of POTS models, of Eve models. But there are like a few limitations to this. And the first one is that the two steps of retrieval and prediction, they are decoupled. And so you first have to figure out like what is a sensible set of sequences that is relevant to the problem that you are focusing on. And sometimes it's a bit tricky. There's lots of recipes to figure out what is the right MSA to put together. And then once you've done this, then like the MSA is fixed, and then you train a model on top of it. And so that feels a bit like not principled, and it feels like there's room for improvement. And the second limitation, I would say, is that these approaches to retrieve MSAs are slow. There's been lots of progress recently to speed them up, in particular for efforts that require creating MSAs for thousands, hundreds of thousands of protein families. And there's been lots of progress in the retrieval literature, more in the NLP field that made some advancements to speed things up. And so that leads me to talk about the Retrieval, which is our proposal to sort of make progress along the two issues I was mentioning earlier. There's lots of things going on this slide, so I'm going to review those different blocks one by one. But at the high level, what you can see is that in the architecture, we have a retriever module and a reader module. This is terminology that comes from the NLP literature. The retriever, the task of the retriever is to identify from a target database the sets of sequences that are the most relevant for the reader, and the reader is going to make prediction based on the context of those sequences. And we want everything to be end-to-end. So we have a hybrid loss that is going to simultaneously train the reader model, so that could be your typical protein language model. The architecture is quite flexible there. And the other part of the loss is going to optimize the retriever, so entice the retriever to retrieve sequences that are most informative for the reader model. So let's focus more on the retriever for a brief moment. So the way we do this in practice, so at initialization of the problem, we first we use an existing protein language model like ESM as the initial parameters for what that retriever should be. We are going to embed all of the sequences in a database of interest. So in this case, we use UNIRF50. So we get for each sequence a compact representation, so mean pooled embedding for all of those sequences. And the advantage of doing this is that once sequences are represented as dense vectors, we can do a very fast search in those databases. And there's been lots of innovation in the NLP literature to support those fast lookups in vector space. So we use some of those innovations, such as this, such as inverted file index. So that's a way to sort of cluster the different embeddings. And when you do a search, so you do a k-mean clustering of your database, and when you do a search, you're going to search first through the centroids of the different clusters, and then fine-tune your search by looking at all of the sequences in those relevant clusters. We use product quantization, which is a way to further compress the information from the dense embeddings, and that allows us to do search very, very fast, like in a fraction of seconds within databases that can be potentially massive. At train time, we are going to update the weights of those retrievers, so they are initialized to ESM, but as the training progresses, the parameters are going to update themselves and deviate to be more useful for the reader that I'm going to talk about on the next slide. The update itself is something that we do infrequently because that's quite costly to recompute the index. And the loss function itself, it was conceived to maximize the relevance of the document that are passed to the reader. So mathematically is the formulation of that. You want to maximize the likelihood that homologous sequences are going to retrieve based on an input query sequence. So that's P retrieval of D. So D is represents a document. That's also terminology that comes from natural language processing. Conditioning on Q, and Q is the query sequence of interest. So you want to maximize this likelihood, the likelihood that the document is going to retrieve given a given query. And the second probability is the one that comes from the language model, that is the likelihood of the query given the document. So you want the document to be maximally informative for your language model. And that's the second half of the model is a reader. So we use a seek-to-seek architecture. So that's an architecture that is going to take as an input the set of homologous sequences and make predictions conditioned on that set. It can be quite costly to do that naively. And so if you looked into the complexity of the attention mechanism, it's quadratic in the length of the input sequence. And once you start having many homologous sequences in your input, it can be computationally intractable to do that. So we use this trick that was introduced in a different paper by Isakar and Grave that is called fusion in decoder. And so like the intuition is that you're going to simultaneously embed those different sequences separately. You concatenate those embeddings, and then you just do a cross-attention between that concatenate and embedding and the query sequence of interest. And by doing that, you've reduced the computational complexity significantly. And the loss to optimize this network, like it's a standard cross-entropy loss. So that's super robust to during training. And as I alluded to, this is just one proposal for this architecture. We actually experimented with lots of different alternatives. You could, for instance, this model that is very nice called POET that also reasons over sets of homologous sequences, and we've adapted this same framework to POET as well. And so the benefit is, so looking at the performance on protein gems, so there's a clear boost in performance, similar in the same way here from Retrieval. And interestingly, so like in terms of performance, if you were to plug sequences from, like let's say Jackhammer into the reader model, so the conditional protein language model, you would get very good performance, comparable with what we get with a trained retriever. The two are performing well in different situations, so there's potential synergies, and we're working on improving that performance further. This is just I'm really excited about this work, because like it's early stage, but you already see the premise of something that could be very powerful. And I think something that is undeniable and like the clear advantage of the approach right now is that it is extremely fast, like orders of magnitude faster than other approaches. So what we looked at was MMSeq and the recent MMSeq GPU version that was itself introduced a couple months ago. That is itself orders of magnitude faster than BLAST, and Jackhammer would not be we would not be able to represent it on this chart. It would be like 2 orders of magnitude longer to do this to collect those MSAs. So like the time per query is 3 or 4 orders of magnitude smaller compared to methods like Jackhammer. So that's super compelling once you're going to train a model that reasons over sets of homologous sequences across thousands or millions of different families. And in terms of next steps, so I alluded to the flexibility of the architecture a few times. So we focused on fitness prediction in this work. There's lots of exciting work that is coming. I think it's very easy to extend those architectures to do structure prediction, for instance. And there are like many improvements possible with other ideas that were introduced in the NLP literature but could be adapted to biology and to protein sequences in particular. So I'm going to shift gears now and not talk about retrieval or zero-shot approaches, but I'm going to focus more on approaches that use labels from computational scanning assays to further boost the performance. And so when you design proteins, you have a choice between 2 types of paradigm. Paradigm number 1, you have an external procedure that is going to suggest mutants, and that could be an exhaustive enumeration of single or double mutations. You can use rational design, so you have an understanding of the protein and the particular residue that are important, and you're going to combine different mutations at those positions, or you can create chimeras. But like the basic point is that you have a long list of protein sequences that you're interested in scoring, and then you have a separate model that is going to evaluate those that list of prospective mutated sequences. The other philosophy of approaching this problem is to distill the information from the problem that you're interested in into the model. And so that gave rise to a diversity of models. I'm going to talk about the first one called Protein NPT and a new version, a new extension of that model in this talk. But there's been fantastic work in the field, such as conditional VAEs that have been more applied to small molecules, but could be applied also for proteins. Guided diffusion, there's a number of work that use that, and reinforcement learning on protein language models. There's a talk from Filippo later today, and I think that's a super exciting area of research that we are at the beginning of it, and it's going to lead to many progress in the field. So let me tell you a bit more about protein NPT. So that's a model that is going to learn simultaneously a joint distribution of sequence and function. By the way, if you were part of the workshop yesterday, I covered this slide and the next couple, but I sort of need those slides to build up to the new work, so bear with me. So the architecture is as follows. So you want to, in the same model, reason over sequences and labels. So you're going to input a mini batch of sequences with annotations into this architecture. The sequences are going to be embedded into a latent space with a sequence embedder. You can use any off-the-shelf language model to do that. And you have a target embedder that is going to embed the annotations into the same space. Once you've done this, during train time, you're going to randomly mask out some of those residues and some of those labels. And the task of the neural network is then to unmask and to predict those masked values. And the advantage of doing this, or the intuition for why that might be helpful, is that afterwards you're going to be able to plug in new sequences and predict their properties. So there would be a new protein for which you don't know the phenotypical value. You would be able to predict with this architecture. And conversely, if you do have a value and you want to make mutation in a particular way, you could condition on those values and sample from the model. So the way it is trained is by a hybrid loss that is going to balance those two objectives in practice. And so if you look at further down in the middle of the diagram, we have those protein entity layers that are going to apply actual attention, which is a method developed in the computer vision literature to reason to the object. And that's, once again, to reduce the complexity of the attention mechanism in those contexts where the input object can be very large. And the way it's typically done is you're going to first do raw attention. So you're going to look at very similar to the standard attention. So for each row, you're going to look at the dependencies between the residues and the property of interest. Then once you've done this, you go through column attention. And so at each position, you're going to look at the similarities between residues across the different sequences. And that forces the network to look at similarities across the mini batch, and that empower the model to have two modes of inference. It can reason between the dependencies between the residues and the property of interest, but also when making prediction, look at similar protein sequences, and if you do have a label for those homologous sequences, make prediction based on those similar sequences. So that's what's happening at train time. At inference time, it's pretty much the same thing, except that we don't mask any of the residues in the sequence, and we only make predictions for the masked target labels. Something that I didn't mention so far is we also have those in purple here, auxiliary labels. And so those could be any attributes that you have about the proteins that could be supporting in making the prediction. So in this particular paper, what we looked at was zero-shot fitness prediction from protein language models that are readily available for all of those mutants. But you can also adapt it to settings where potentially the target that you want to predict is low throughput, high quality data, and the auxiliary label are high throughput. You have lots of them, easily accessible, and those values can help you predict the low throughput label annotations. And this is to stress once again on the ability, or like the philosophy, why we went down this track of learning joint distribution of sequences and function, is that once you do have this, once you have learned this joint distribution, then you can condition on things and sample on the remainder. So you can condition on high value of the property that you're interested in. Let's say you want to take an enzyme and make it more thermostable. You would set the value of the label to high thermostability and ask the model to mask a few positions in the protein sequence, and ask the model to sample new variants at those positions. And the sampling process will be such that those new mutants are more likely to be connected with that condition on high thermostability. And so in practice, so in an in silico experiment that works very well, we are currently working on testing that in the lab. And before I talk about that project in the wet lab, I wanted to mention this effort called Primo, which pushes that protein NPT architecture to the next level. So in protein NPT, we train one model per protein family and assay, so one model per assay really. And the idea there was, what if we were pre-training that model across multiple assays at the same time? With the hope that if you do this type of pre-training on all of the high-throughput assays that are currently available, you could then more quickly generalize to new situations, so new assays for new proteins. And potentially be able to predict the phenotypes that you're interested in with much fewer annotations. And so that's what we are doing here. So like as the input of the architecture, we have several assays that we process. There's like a few core ingredients to the architecture. So the first one is that we have separate embedders for different properties to tell the network that when they are learning a distribution between sequence and function types, like it matters if that function is about thermostability, about catalytic activity, or binding, etc. So like that's the role of that property type embedder. Another modification compared to the prior architecture is in the attention mechanism to adapt the architecture to indels. And so, like in the interest of time, perhaps I'll skip those technical details. Preference loss is a core aspect of the architecture to make sure that we are able to learn across different phenotypes of different nature. So, like different assays of measurements that are on different scales, and to be able to facilitate the extrapolation to those different settings. It was easier, we felt like it was easier and it worked better in practice to use a preference loss, so that's what's described at the bottom of the slide. And other works had already shown that this was favorable in other settings. And the last ingredient is test time training. So what we find is very useful in practice is let's say you've pre-trained that primer architecture on a bunch, let's say hundreds of assays from Protein Gym or MaveDB, and you want to then use it for a net new assay. The way you're going to use this model is you're going to feed in a handful of labels. It could be 4 or 8 labels, and the concept of test time training is so you could make prediction as is. So you plug in those labels, and because the model was pre-trained and has been exposed to multiple examples of sequence and annotation, is going to be able to generalize in context with those few annotations without changing anything. But we do find that it is helpful to do this test time training, which is an approach that has been proposed in different parts of the ML literature for different tasks. And it's very useful. So showing you the results here So the top half is on protein gym assays that we have left out. So we train on a bunch of assays and we evaluate on assays that have been set aside. And the architecture works very well in even with very few labels. So that was a bit of the limitation of prior approaches. You know, what do you do when you want to learn from 4 labels, 8 labels, etc. I think that this pre-training is very good at giving you like a good starting point to leverage those few labels. And the test time training is super important when the fitness landscapes are rugged. So that's when we look at the DMS level performance within the protein gym assay, we do see that. And in the bottom half, what we did is to put together another set of assays, and not from protein gym. Those assays are they are evaluating the landscape of natural protein sequences. So you have, for instance, hundreds of different measurements for correspondent mutase that they are all natural sequences, and they tend to be farther apart from one another compared to the sequences in protein gym. And what we do see in those instances is like it's super important to use this test time training to boost the performance, to allow the model to adapt to landscapes that are quite different to the ones that the model has been exposed to during training. I may have like a couple of minutes. So I briefly mentioned the application that we are working on. So we're working on carbonic anhydrase for DAC systems. We want to make it more thermostable, function at higher pH, and functional in industrial solvents. And for this, we are using the tools that I just introduced. It's a collaboration with the Savage Lab and the Baker Lab, where we all focus on different methods. So the Savage Lab has developed a high throughput assay that we use as part of this 3-step approach that we use to filter lots of designs that we come up with various approaches. Some of those approaches are based on alignment-based approaches, other based on inverse folding and protein language models, and a few de novo designs from the Baker Lab with our recent RF diffusion model. So we use this high throughput assay to filter, and in the last step where we are right now, we use those models that combine sequence and function in the same model. So to learn from the measurements that we do in low throughput. So like in the first step, we have this high throughput screen to evaluate thousands of different mutants, and then in the second part of the project, we've been focusing more on the low throughput assays that more precisely measure the stability and the pH resistance, so the activity at different temperature, the activity at different pHs, to test out the functionality and the conditions that the protein will be exposed to in the bioreactor. So we are in that last step, and we hope to share those results very soon. So we'd like to thank everybody for your attention, and many of my collaborators that have been involved in the various projects that I presented, Deborah Marks and Yarin Gal, that have been a supervisor during my postdoc and PhD, Johnny Mafala for the involvement in protein gem and transception, Aaron, Danielle, and Louis, and many others rose in the room to be that were the powerhouse behind the protein gem effort. Ruben and Peter that have pushed the Retrieval model, and Felix and Sam that worked on Primer. Thank you very much.